Hello viewers, so today we're going to learn about physical agents to control food contamination. Well, there are three types of contamination: biological, chemical and physical, and today we're going to learn about physical. Let's begin. Well, starting with what is physical contamination. As I told in the introduction, there are three types of contamination. Well, physical contamination is when harmful objects contaminate the food, it leads to physical contamination. For example, what are the physical objects which are usually considered as contaminants, like hair, rat, pests, glass, metals, which contaminate the food, or plastic wrappers, etc. So usually when you are consuming any food, and if these kind of physical objects are present in the food which cannot be consumed, are usually considered as physical hazards or physical contaminants. Hello everyone, so as you can see from this current slide, these are the physical agents which are usually used to control the contamination and make the food sterilized and edible. So there are two main agents, heat and radiation. So this is the main basic flow chart which will be really easy for you to understand. First, coming to heat, there are two types of heat, dry and moist. Dry heat is incineration and dry oven. In dry oven, it leads to sterilization where dry hot air is released and all the microorganisms or any physical agents which are present will be sterilized, and which will be usually eliminated. And coming to moist heat, there are two types again, steam under pressure, and one more is boiling water, hot water and pasteurization. So here to be noted that boiling water and non-ionizing UV only causes disinfection, others all will lead to sterilization. So there is a slight difference between disinfection and sterilization, which I'll be telling in further videos. Well, coming to steam under pressure, that is autoclave is used, where steam under pressure is happens and sterilization of the physical agents is done. And coming to radiation, there are two types of radiations, ionizing and non-ionizing. Ionizing uses X-rays, gamma rays, cathode, and non-ionizing uses UV and disinfection. In the coming up slides, I'll be explaining everything in detail. Coming to moist heat, which I've shown you, moist heat comes under normal heat, and normal heat is of two types, dry heat and moist heat. Well, coming to moist heat, as I told you, autoclave is used. So this is the picture of an autoclave where steam under pressure is used. The steam sterilization, the autoclave is also called as steam sterilization, which is a type of moist heat method which is done by using instrument, namely autoclave. It is formed due to the boiling of water at a temperature of 100 degrees centigrade, where water is used at 100 degrees centigrade, and at 100 degrees centigrade the water boils and steam forms, which is in the vapor form, which is not released out and which usually gets trapped. So these you can see the knobs. These you can see the knobs which usually does not allow any heat or air to come in or go out, and hence the vapors are trapped inside, and due to this the steam which it gets in contact with the material, the food material or any material, it kills the organism which is present on it by entrapping into the cells of the organism and degrading the cells. Well, this steam which gets in touch with the material, it kills the organism by transferring the heat. The vapor which contains the heat is transferred into the microbe, and hence the microbe cannot tolerate that such high temperatures, hence it is killed. So coming to dry heat is also a subdivision of heat. Well, dry heat, hot air oven is used. Hot air oven, it leads to sterilization. This is also called as dry heat sterilization. In this, there is no moisture or water used, so there is no vapor entrapment, unlike autoclave. Here, hot air oven is also called as dry heat sterilization. It works on a principle of heat conduction. So as you can see, there is a tray over here. So on this, the product is kept, and this is the doorknob which is locked, and the heat, it passes to each other, and there is no water to be noted. So the principle it basically works on heat conduction, where the food product is sterilized layer by layer from starting to the ending of the surface. Suppose for example the food is kept over here, layer by layer the heat is passed through the food product, and layer by layer the food product is sterilized. Well, the temperature of this hot air oven is varied, like from type for the food product. There are different temperatures they are used. So usually from 50 to maximum 300 degrees Celsius is used to sterilize completely the food product. So from the continuous heat flow from the blowers, there's a blower over here, and there's a continuous heat flow from the blowers, due to which there's a continuous heat rotation, and since the door is locked completely, it is double walled insulated, so the hot air cannot be escaped outside. So there's a continuous heat flow rotation within the hot air oven, which makes sure the heat is entered into the food product, and it is sterilized. And one more point to be noted, that the trays in the hot air oven, which are usually of aluminium, and the doors are double wall insulated, and these aluminium trays is also acts as a heat conductor. So coming to radiation, so as I told you, for physical agent control, there are two types, heat and radiation. Heat, we have read two types, dry heat and moist heat. So now we are going to study about radiation. Here in radiation also there are two types, ionizing and non-ionizing radiations. So coming to non-ionizing radiations, radiation in sterilization, it relies on ionizing. Primarily, ionizing radiations consist of X-rays, gamma rays, uh, and other radiations. So coming to here, the sterilization, what are the advantages, what are the basic advantages of radiation? Here advantages is usually gamma rays or X-rays or primary gamma rays which are used, it can easily penetrate through the food products, and it can permit the heat sensitive materials. That means there are certain food products which are heat sensitive. That means if they are heated at high temperatures, they might spoil. So radiation is used in that, and these food materials, these radiations, they actually penetrate even to those sensitive food materials, and they can kill the microorganisms and sterilize it. So it is an instantaneous and simultaneous sterilizing effect. This method is very instant, and there are numerous advantages over heat and chemical based techniques. So your point to be noted is that the heat, the dry heat or moist heat, cannot be used in all circumstances. That is, in all types of food at all seasons, the heat method cannot be used. So since because of that, there's a backlog. So radiation method has come into phase, and ionizing and non-ionizing is being used. So coming to disadvantages. First of all, radiation is very dangerous, so it should be done only by a well-trained, experienced staff members. It's not a all person type of thing that everyone can be done. It should be either the person should take a proper training, or a well-trained staff should be doing it, and the training is continuously given to all the sub staffs also, so that the workload can be divided. So coming to this radiation, while the radiation is done, you cannot just leave it and go somewhere else. You cannot do multitasking. So there should be continuous monitoring of how the radiation is doing. Is there is too much of radiation or there's too less? It should be balanced, right? So there should be continuous monitoring and specially designed purpose. That means a special hall, a special room should be designed in such a way for radiation only. There should be no other equipment, because since it's a radiation and it's a very big thing. So in the previous slide, we read about how the ionizing radiations, it penetrates through the food materials and sterilizes the food materials, and hence it can be consumable. So here we're going to learn about non-ionizing radiations. The basically non-ionizing radiations, the first one which comes into mind is UV radiations. So why is non-ionizing radiations basically used? There is a primary question, right? Because since we have heat and radiations, so there are certain food products which I would love if you name them in the comment section. That there are certain food products which are really heat sensitive, but they do not need that much of penetration of the radiation, so they can be easily disinfected from non-ionizing radiation itself. So basically it means that when the work can be done by non-ionizing radiations and with less penetration, with less energy, why to go towards high energy, high end radiations? So basically, industrialists, so scientists, they first prefer non-ionizing radiations. If the food is getting disinfected and sterilized by this, it will be done by non-ionizing. If not, if they need more energy for the food to be sterilized and disinfected, they go towards the ionizing radiations. So coming to UV lights, the UV radiation, they're less penetrative, they have less energy, and they can be controlled, and they're unidirectional itself. They are made unidirectional, and the wavelength is of 250, which is very toxic. That means basically UV radiations is used for bacterial spores and other vegetative cells, because their wavelength of 250 is more than enough for bacterial spores and vegetative cells to kill. Commercial UV lamps are also used in the market to use as a disinfectant. UV lamps, the picture is over here, which these are basically used for the food processing. So bacterial spores and vegetative cells, if they can be killed by non-ionizing radiations and UV radiations, then why should any industrialist prefer ionizing radiations, right? Because they are high-end energy radiations. So that's it, people. We had learned today about physical agents to control the contamination in the food products. We read about the heat and radiation. There are two types. What are physical contaminants and what are ionizing radiations, non-ionizing, dry heat, moist heat, etc., etc. So these are my references, and with this I conclude. Thank you everyone, and please do like, share and subscribe, and please do share to every food technology student, share to every nutrition student, and thank you very much.